This lecture is on blood types. I'm Dr. Tandra Chakraborty, the instructor for this course. We are continuing with the blood chapter where we learned about blood, composition of blood, function of blood, the, uh, the life cycle of blood, and then we also learned a little bit about the recycling of the blood, or mainly red blood cells. Here we are learning about blood types. So one of the most important thing to understand here is we are actually going to learn about blood types, and the reason we are going to restrict it to only certain type of blood types is because those are the most important ones, and we are going to just learn a few that are very important or present in the population. So, before we go into the different kinds of blood types, let's understand three main, I would say, topics or words. Number one is antigen. Antigen are substances. These are complex molecules present on the surface of the cell. They actually are responsible to trigger an immune response. Okay, so on the surface of the cell, any cell, blood cell, of course, red blood cells, there are any red blood cells have these surface molecule which are known as antigen, and they trigger an immune response. These are surface antigen substances, and they are able to recognize as normal or self. And in other words, the immune system ignores these substances rather than attacking as foreign. So they know, our body knows, these antigens that are present in our red blood cells are our self cells, and they are not considered as foreign cells. Antibodies are present in the blood plasma. So if you remember, blood consists of two parts. Number one is formed elements, which are the red blood cell, white blood cell, and platelets. Here we are just talking about red blood cells. And then there is plasma, and the plasma has these antibodies, which are basically globulin molecules that are present in the blood plasma. They are part of the immune system, response to foreign matter. They go and attack. And whenever there is a foreign antigen present, they'll go and attack them. So one important factor to remember, these antigen and antibodies, if they are similar or same, it causes, they bind to each other. And when they bind to each other, they cause clumping of the red blood cell, which is not good. Okay, when the cells clump together, it is not okay. It is not good. So they will destroy the cell if they join together. Okay, so these, if they are from the same cells, we will talk about antigen and antibodies very soon. In the next slide, you'll understand it. So, but just remember, antigen and antibodies binding together in the blood causes agglutination, and that is causes clumping of the red blood cells, which is basically lysis or destruction of the red blood cell. So if you look in here, we are talking about four different kinds of blood types: type A, type B, type AB, and type O. Type A, this is the red blood cells, and on the surface shown in purple is the antigen A. Again, type A red blood cells have antigen A. Type B, shown in green, are antigen B. Type AB has both antigen A and B. And type O does not have any antigen. I want to emphasize the picture shows purple, green colors and circle and, squares, but actually they are not as such. So you do understand these are protein molecules. These are just shown for you to understand. The next part is, so we learned antigen A has, type A has antigen A, type B has antigen B, type AB has both antigen A and B, and type O does not have any antigen. Now in the plasma, And what antibody does type A have? It has antibody B. Type B has antibody A. Type AB does not have any antibody. And type O has both antibody A and B. Okay. So this information is so important for you to understand that going forward, if you understand this, this is going to help you to understand rest of the lecture. Apart from these antigen and antibody, there is another factor which is known as Rh factor or rhesus factor. If the rhesus factor is present, it's called positive. If the rhesus factor is not present, it's called negative. Okay, so if you have it, it's positive. If you don't have it, it's negative. Okay, so let's go to the next slide. And this will help you to understand it better. So if you see type A, this is the red blood cell, and on the surface is antigen A. What antibodies it has? It has antibody B. Okay, so antibody B. Type B, on the surface, it has antigen B, and the plasma, it has antibody A. Type AB has both antigen A and B, and it has none of the antibody A or antibody B. Type O does not have any antigen. So in the plasma, it has both antibody A and B. Why is this arrangement? Okay, why is this arrangement? Think of a situation where this red blood cell with antigen, let's take this example, this red blood cell with antigen B as shown in this figure. Okay, this is red blood cell uh, type B. This is type B blood group. It has antigen B on the surface, and if the plasma had antibody B, I'm just giving you a hypothetical situation where it's having in the plasma antibody B. This antibody B will bind to the antigen B and then cause lysis of the cell. And that's what is called hemolysis or agglutination. So, never in the blood will antigen A, red blood cell with antigen A, have antibodies for A. It will have antibodies for B, but will not have antibodies for A. Why? Again, if it does have antibodies for A, it will cause clumping and destruction of the cell. This is the reason. This is the reason it decides which blood a person can receive and which blood a person can donate. Remember, there are many, many circumstances. It can be a disease, it can be an accident where blood transfusion is required. When a blood transfusion is required, one thing to remember is the blood that is given to the person who requires it, who needs it, the blood that is given can be divided into red blood cells and plasma, and only the red blood cells can be given. The plasma is not given. But the receiving person who receives the blood has the entire blood. It has the red blood cell with its antigen and also has the antibodies corresponding to it. So, so for example, in red blood cell A, for example, red blood cell A has antigen A. So it can only receive blood from type A person because it has antibodies B. It cannot receive blood from type B. On the other hand, since it has antibody B, it cannot receive blood from type AB because if it does, these antibody B will bind to these antigen B and coagulate. But it can actually take up type O because it does not have any antigen on the surface. Next, let's take the type B. Type B can receive blood from type B. It cannot take up type A because it has antibody A, which will bind to the antigen on the red blood cell and destroy it. It cannot take blood from type AB because this has antibody A, which will bind to the antigen A on the surface of AB. It can take up blood from type O because it does not have any antigen on it. Type AB, since it does not have any antibody A or B, can take up type A, type B, and O. Type O, type O does not have any antigen, but it has antibody A and also antibody B. So it actually cannot take up blood from any one of them. So type O is known as the universal donor because it can give blood to each one of them. And type AB is known as the universal acceptor because it can accept blood from any one of them. In addition, there is another factor which is known as the rhesus factor, Rh factor. If it is present in the blood, it is known as positive blood. And if it's not present in this blood, it's known as a negative blood, Rh negative. So if it is present in type A, we call it A positive. If it is present in type B, it's called B positive. If it is present in type AB, it's called AB positive and O positive. If it is not present, it is called A negative, B negative, AB negative, and O negative blood. So this is what we were learning about, the Rh factor. It was first discovered in rhesus monkey, and that is the reason it is called the Rh and positive if it is present, negative if it's not present. So one thing to remember here is the Rh positive, when there is rhesus positive antigen present in a cell, the plasma does not have any antibodies. But if it is a negative, if the cell does not have rhesus factor, then it does have antibodies for rhesus factor. So there is a problem that happens during pregnancy because of the presence of this factor. And we'll learn that eventually. Before I do that, let's understand blood typing a little bit, and then we'll go ahead with that. So this is blood typing, blood type sample in each one of them. If there is agglutination form, that means it has this is a plate where agglutination has happened. So that this is antibody A. So it must have been given antigen A in order for it to form agglutination. So this is blood type A. There is no effect. And since again agglutination has happened with anti-D, which is rhesus factor in here, so it's A positive blood group. Again, in this agglutination has happened. These are plated with antibodies, so must have got in antigen B. As a result, this is B, and since rhesus factor is also present, this is B positive blood. In this case, there's agglutination in A and in B, and rhesus factor. So this is AB positive. And the last, since agglutination has not happened in any one of them, it is O, and rhesus factor is also not present, it is O negative. So this is the next phase, the one that I was talking about, that during pregnancy there is a disease which is known as hemolytic disease of newborn, HDN, or known as erythroblastosis fetalis, happens during the fetal development. Okay, so the first pregnancy is likely if the rhesus woman, which carries an Rh positive, you can see this is Rh negative mother carrying Rh positive fetus. The first pregnancy is likely to be uneventful, or nothing happens, because the plasma normally prevents the maternal and the fetal blood from mixing. And the plasma is there, amniotic sac, so it does not let it mix. However, at the time of birth, or if there is a miscarriage also, there is tearing of the placenta and mixing of the blood. She now, the mother, now starts making antibody for the rhesus factor. So, when she again has a child which is Rh positive, again, if the child is Rh positive, then those antibodies that was developed during the first pregnancy will attack the child. Agglutination of red blood cells hemolyze, and the baby is born with severe anemia. Okay, this is called hemolytic disease of newborn, or erythroblastosis fetalis. So this happens 1 in 10 cases of HBO. Incompatibility, it really is not a very common thing. And now we have diseases, we have medicines that can prevent this from being fatal also. So, but it does occur. So it's essential that the mother's blood group and the fetus's blood group is checked during the entire process of the pregnancy. So as I said, Groham is given to pregnant Rh negative mother uh, so that the entire process can go on well. This actually concludes the blood typing. Just to review it a little bit, we mainly have 4 types of blood group that we want to talk about. One is type A, B, AB, and O. Type A has antigen A, antibodies B. Type B has antigen B, antibodies A. Type AB has antigen A and B, antibodies for A and antibodies for B. Type O has no antigen and has antibodies A and B. AB is an universal acceptor of blood. O is an universal donor. There is another factor which is known as rhesus factor or D factor. If it is present, it's called positive. If it's not present, it's called negative. Or we just don't say anything. Positive or negative, we can call them, or sometimes we don't even mention it. This actually ends the blood typing and cross reactions.